I'm going to start by covering the activation of B cells. We've already talked about the activation of lymphocytes in general. Hopefully this will be review for you. The first thing that has to happen is the BCR, the B cell receptor, has to bind with the antigen. When the binding occurs, this initiates receptor-mediated endocytosis where the antigen is brought into the B cell. Once this occurs, the B cell begins to divide, multiply, and this is called proliferation. The cells will differentiate into two types of cells, effector cells, which are your plasma cells, and these are the ones that will be involved in eliminating the pathogen or the antigen, and they produce the antibodies. In addition, you'll be producing some memory cells, and memory cells are going to provide protection later on if you are ever infected with the same antigen. We'll talk about this role a little bit later in this video. Recall that all of the daughter cells after this proliferation will have exactly the same BCRs, which means that they will recognize and bind with the same antigens. Therefore, you're creating this army against that specific antigen once activation has occurred. B cells typically require the help of helper T cells, and we'll look at how this occurs in just a minute. But helper T cells will release interleukins. Recall, these are a type of cytokine which will go back and bind to the B cell and help it finish its activation process. In some cases, you might have a large antigen that does not require the interleukins from the helper T cell to be completely activated, but most B cells will require helper T cells to complete their activation. This figure illustrates the role of helper T cells in activating the B cells. This is your B cell. This B cell has already bound to its antigen and brought the antigen in. Once the antigen is brought in, that B cell is going to bind that antigen to an MHC. Remember the major histocompatibility complexes, these proteins here. So it will bind the antigen, which is the red structure, to the MHC. The MHC in this case is going to be an MHC class 2. MHC class 2s are recognized by CD4 T cells, and CD4 T cells, when they are activated, become helper T cells. Also recall that the CD4 is a protein that is bound to the plasma membrane of the helper T cell, and that protein binds to the MHC here to complete this binding that occurs. As soon as this happens, the helper T cell will release interleukins. They will diffuse across the interstitial fluid and bind to the B cells, so you'll have some receptors on the B cells for the interleukins. Remember, receptors are always proteins, and when the interleukins bind to the receptors, that triggers the B cell to complete its activation, begin proliferation and differentiation.